Good morning to you all. I welcome you all for another session of discussion on the concepts of bioprospecting or the gene prospecting. It is an important concept which we have been discussing under Human Genome Research or project. If you just look at the workflow of the bioprospecting, it begins with genome sequencing, followed by that is the assembly, and then the gene prediction, annotation, and finally data sharing. So in the last session, we were discussing about the second and third generation sequencing methods. So we had discussed about the Illumina, ion torrent methods under second generation sequencing methods. Then we were discussing about PacBio sequencing method and about ONT, that is Oxford Nanopore technology method under third generation sequencing methods. And I did show few bites about the working principle of each of these methods. Following the genome sequencing, in the workflow of bioprospecting, we have genome assembly. We were also discussing about the genome sequence assembly and the algorithms being used for the process of assembly. In today's session, we will discuss further about the bioprospecting or genome prospecting. We need to discuss about gene prediction, gene annotation, and data sharing. There is one more concept called binning. If you just recollect the workflow which we have discussed in the last sessions, binning and assembly, they happen together so that the gene prediction process becomes easy, right? So following sequencing and assembly, we have the binning, that is phylogenetic binning. It is about phylogenetic analysis of the taxonomic groups present in the sample that is being analyzed. Right? A phylogenetic analysis of the taxonomic groups that is present in the sample being analyzed. Binning is the process of clustering genome sequences into groups so that each subset represents a separate biological taxon. So what is happening in the binning process or the phylogenetic binning? See, we have sequenced. Now we are moving to the process of assembling the sequence, and then in between we have the phylogenetic analysis of the taxonomic groups. We call it as phylogenetic binning. So it is a process of clustering genomic sequences into groups, and then each subset, it represents a separate biological taxon. So what you have to remember here is the process of clustering genomic sequences into groups. So binning and assembly are two related procedures. They are not unrelated or totally independent procedures. They are related procedures. So binning can be performed prior to the assembly of the genome sequence, or it can be integrated in the process of assembly itself. That means when you are carrying out the assembly, you can integrate the process of binning into the assembly itself. That means you have the sequence, following the sequence is the genome assembly, and binning can be performed, that is clustering of the genome sequences or phylogenetic analysis of the sequence. It can be performed prior to the assembly, that is pre-assembly procedure, or it can be integrated into the assembly process. Binning and assembly are two related procedures, so that it can be performed pre-assembly or integrated into the assembly process. Now, what is the advantage of the binning process? In either scenario, whether it is pre-assembly mode or integrated mode, binning always attempts to prevent co-assembly of mixed genomes. It never mixes the genome sequences. It always prevents co-assembly of mixed genomes. In either scenario, in either case, whether it is pre-assembly mode or integrated into the assembly process, in either scenario, binning attempts to prevent co-assembly of mixed genomes. Is that clear? We have the sequence, followed by the sequence is the assembly, and we have something called as the phylogenetic analysis or the binning process. And the binning and assembly are two related procedures, and binning can be performed pre-assembly of the sequence or can be integrated into the assembly process. But in either situations, the binning attempts to prevent the co-assembly of mixed genomes. Is that clear? If you just look at this image, you will find that this is called as taxonomic binning or the binning process. So what is happening? You have the samples, then you have the sequence, and then you are in the process of assembling, and this binning process can be integrated during the process of assembly, or it can be even in the pre-assembly mode. That means you are clustering the genomic sequences into groups based on some of the taxonomic features. Is that clear? So in theory, each bin represents a single genome. You have to be very, very careful. Each bin represents a single genome and is assembled separately. We know that what is binning? Binning is genome clustering into groups. Then what does each bin represent? Each bin represents a single genome and it is assembled separately. So if you just remember the discussion we had in the last slide, in either scenario, binning prevents co-assembly of mixed genomes. So here what we are trying to understand is each bin represents a single genome and it is assembled separately. So there is no co-assembly of mixed genomes. It also removes some of the problems of incorrect assemblies, connecting contigs and diverse taxa. So never ever it allows co-assembly of mixed genomes. It prevents co-assembly of mixed genomes. Got it? It also removes some of the problems of incorrect assemblies which would have arisen due to diverse taxa. Right? So we have understood about the sequence, we have understood about the assembly and also about the binning process. Then what is the algorithm being used for the binning process? Likely bin. Likely bin is an unsupervised statistical approach for binning metagenomic fragments. This method uses Markov model or Markov approach. So what is the basis for this approach? It is built on the assumption that the oligonucleotide frequency distribution is homogeneous within a genome or bacterial genome. That means the oligonucleotide frequency distribution is homogeneous throughout the genome. That is the basis for Markov approach or Markov model. So likely bin, it is an unsupervised statistical approach for binning the metagenomic sequences or fragments. And this method is reported to perform well in low complexity metagenomic sequences or communities. So the genome sequence should be of low complexity, only then it is possible to use the likely bin approach, which is based on the assumption that the oligonucleotide frequency distribution is homogeneous within the genomes or the bacterial genomes. Likely bin uses Markov model. The assumption is oligonucleotide frequency distribution is homogeneous and it performs very well when the micro organism genomes or the metagenomic communities have low complexity. Is that clear? So we have seen sequence, followed by that is the assembly and then the binning. Next step is gene prediction, that is identification of potential genes. It is all about annotating the assembled data and identifying genomic features such as genes and regulatory elements. So what we do in gene prediction? Identification of genes and regulatory elements. Fundamentally, it is the identification of potential genes. So the short reads produced by the sequencing methods, that is NGS, could be second or third generation sequencing methods, they are difficult to be assembled, and even if you try to assemble, the context and scaffolds can often be short and fragmented. So you need to have a robust algorithm. So meta-gene annotator, it is a metagenomic gene finding algorithm which predicts genes on short sequences from an uncharacterized metagenomic community. We are discussing about prediction of genes, that is identification of potential genes, and the widely used algorithm is the meta-gene annotator, which is a gene finding algorithm. What is the basis for this meta-gene annotator? It predicts genes on short sequences from an uncharacterized metagenomic sequences or the communities. Now the assumption is, it is based on the assumption that CG content correlates with dicodon frequencies. Dicodon frequencies, that is two adjoining codons, which is a latest approach in gene finding or gene prediction. Right? Now, the dicodon usage patterns, what does it do? It encapsulates both the locally available information as well as global information about the DNA sequence. It is based on the assumption that the ribosomes actually reside over two codon positions when they slide along the mRNA. Right? So the ribosomes, they are actually residing on two codon positions, that is two adjoining codons, we call it as dicodons. So the frequency of these dicodons are used to predict the gene or understand the gene function. So these dicodon usage has a direct biological rationale. So that is what this meta-gene annotator uses. Right? Next we have Orphelia. Orphelia is available as both a web server and as command line tool, and it uses two-step machine learning approach. Now people are discussing a lot about AI and machine learning. A lot of advancement has taken place in utilizing the principles of AI and machine learning to decode the genome sequence secrets. So this Orphelia uses a two-step machine learning approach, and it predicts the gene function, structure and function. Right? So this is the web page or the landing page of the Orphelia, and a lot of information can be found about this machine learning approach to predict the gene structure and function. Following gene prediction, next step is gene annotation. That is, it is the identification of domains, functions, and metabolic pathways in which the gene products are involved. So there are large number of published protein sequence or the features, that is the features which determine the proteins or the genes structure and function, the structure databases, and each of these have their own strengths and weaknesses. So how to go about gene annotation? We have InterProScan. It is a protein function prediction software which searches the 12-member databases. InterProScan, a protein function prediction software, most commonly used software that searches the 12-member databases simultaneously, so that you can in very short time you can predict the function of the protein of your interest when you have the sequence of that. Then you have Reactome. Reactome is a free open source and curated database of biological pathways. The reactions are organized in hierarchy in the Reactome software, with single reactions in the lowest level, while interconnected pathways are organized in higher levels. We have InterProScan, a protein function prediction software. Then we have Reactome, a free open source and curated database of biological pathways, wherein the single reactions are at the lowest level and interconnected pathways are organized at higher levels. That means there is a organizational hierarchy in the Reactome software. Then we have KEGG, that is Kyoto Encyclopedia of Genes and Genomes, which is a database connecting genomic, biochemical and phenotypical information from multiple individual databases. Then we have Metasys, which is a large comprehensive database of pathways and enzymes. It is the largest collection of curated metabolic pathways software. Right. Then the final step is data sharing. That is, research findings are shared across the scientific community. Right? So how do they do this? They have a beautiful algorithm for that, that is Metaphor. It is a simple web application that allows users to raise a query, to go about search or searching the sequences, annotated sequences, curated sequences, and browse the millions of gene and protein predictions that are the results of metagenomics assembly and annotation. So Metaphor, it is a simple web application that allows the users, it is user friendly to raise a query, to go about searching the genome sequence and then browse for the gene and prediction of the functions of the genes. Right? So Meta4 includes web services to access tools such as BLAST, that is basic local alignment search tool, and InterProScan. The advantage, the beauty of this Meta4 is that it can be set up even on a private or institutional server prior to data release and publication. So it is so much user friendly. You can make use of this Meta4 even on your private laptop or the desktop or the institutional server prior to the release of the data and publication of the data. Right? So this is the illustration about the working mechanism of the Meta4. So having understood about the Human Genome Research or Human Genome Project, what we have learned so far and what the people actually involved in human genome research think about this project and what is their experience is given in a beautiful bite over here. So I wish you people can go through this bite, which is what we have discussed in few of the last sessions. So it's a reflection of what we have discussed. So have a look at this bite.
In 1989, a group of scientists was assembled to decode the human genome under the direction of Dr. James Watson.
One of Watson's big motivations here was to try to recruit the best and brightest from many different disciplines, biology, but also physics, chemistry, engineering, because all of those would have to get together if this was going to work.
Many of the biologists, and I love them dearly, of course, were dragged into it kicking or screaming in the beginning because they failed to see the tremendous value that such an enterprise would provide the field.
The talent turned out to be all over the world, so you have to basically make it possible for those bright minds to get engaged where they are. They're not going to all move to join this effort.
Genomics has fundamentally changed the way we do science. It brought the notion of team science and collaboration to biology.
Curious thing about collaboration, it's also intensely competitive. Each person wants to achieve something personal. You know, they want to make their hit, on the problem, but then immediately they want to share it with the other, and the other learns immediately. And so you ratchet each other up.
From the beginning of the Genome Project, there was a fairly strong sense of enthusiasm in the public sector, the private, and academia.
We had lots of support from Republicans, partly because Newt Gingrich was interested in science, and I knew it. There's no partisan difference in everybody wanting to live as long as they can.
I think there's a lesson there. It isn't a big monolithic project. It was a big challenge, but it brought people together. And I think that's what we have to do in the future.
When the Human Genome Project was started, it's important to note the technology of sequencing was not developed yet. So it's, in a sense, a project that was launched with a wing and a prayer.
We couldn't sequence it at that time. The machines were nothing like fast enough. There were no machines even. It was sequencing was done by hand.
I thought we might have to do it brute force, and I was quietly prepared to use the old-fashioned technology just because it was so important to get it done.
There are a lot of things, that would make life easier that just hadn't been invented yet.
It'd be a 100-year project, like monks working to copy a book at best.
Bit by bit, we got into more and more large-scale sequencing. But the sequencing is enormously important because it provides the possibility of searching by computer.
When I showed up at Washington University one day with a computer terminal kind of under my arm, people looked at it and said, "What are you going to do with that?" This is a genetics department.
Genomes were supposed to be too complex to be able to reassemble them in the computer.
Without computers, we simply would be dead in the water.
Computers and genomics were made for each other. Genetic information is digital. It's like binary code. It just happens to use 4 digits instead of 2. It is amazingly accurate, and that's what biology gets from digital information.
The Human Genome Project contributed a cultural change about data sharing, because the Genome Project was about sharing data almost immediately. And while it seems obvious now, it wasn't obvious then. That required significant effort.
I think I'm quite lucky as a scientist because I grew up always sharing data. The community around me, the genomics community, the worm and fly communities as well. It's just a default that you're going to share everything. You're going to share the code. You're going to share everything by publishing, but you're going to share it before publishing. You're going to share half-cooked ideas. And in fact, this is a wonderful thing. It's liberating.
It would have been virtually criminal to try to hoard discoveries that meant the difference in life and death in people.
This is information which we'll be using for centuries. The sooner we get it and everybody has access to it who wants it, the faster we can move in terms of understanding how life works and how disease occurs.
Patients invest a certain degree of fear about genetics. Who's going to find this out? How will it be used against me?
A lot of the ethical and social considerations that we're struggling with today really have historical roots in the early 1900s in the United States, where we had many states that had laws that allowed us to forcibly sterilize people and prevent them from having children based on whether we as a society felt that they were genetically fit.
So at the beginning of the Human Genome Project, we got it right of bringing in the social scientists, the ethicists, the lawyers, and we now have helped to provide a whole generation of scientists that are focusing on the ethical and social issues of genomics.
The sequencing of the human genome was a monumental achievement. Decades of work, thousands of scientists, billions of dollars, and we get this first roadmap for what makes up the human genome. It's an incredibly exciting moment. At the same time, it's just the beginning of what's really a much longer voyage.
And let's again be audacious, but let's not be unrealistic about what's possible. I think the next 10 years are going to see some exciting developments in genomics, particularly those where we start to use genomic information as part of routine medical care. And that'll be applied to dealing with diseases like cancer, hypertension, diabetes, and Alzheimer's disease.
I dream of the day when every young mother brings a young girl or young boy home from the hospital with a little gene card, and they say, this is the way you should prepare this child's health care. It's exhilarating.
So thank you all.